Good morning. I'm just waiting for the slides to appear, and whilst that is happening, there we go. My name is Cheney. I'm a research fellow and clinical trials methodologist at the Centre for Trials Research at Cardiff University. And I'd like to thank the organisers for the opportunity today to speak a little bit about some of the concepts and challenges surrounding trialling advanced therapies in HD. So what are some of the constraints that we need to consider when we're talking about trials for advanced therapies? As this audience will know, HD is a rare disease, so that means we have fewer number of participants from whom we can recruit. As Liam has just discussed very eloquently, these therapies require direct intracranial delivery. And whilst I'm not going to talk about the challenges of actually getting the therapies into the brain, what I want to mention here is that these trials require some kind of element of neurosurgery and that they are a one-shot deal. You know, once we've put something into the brain, we cannot take it back out again. It's not reversible. And then there's the problem that we have with follow-up. As, again, Liam has just mentioned, it's going to take a long time for us to measure any potential functional benefit of these therapies in people. And when you have a long follow-up period, it leads to complex clinical trials that are expensive to run. So overall, how do we maximise the efficiency of our trial designs to mitigate against these constraints? So that these constraints that I've just described raise some very specific challenges. So we need to think about our overall study design. Which is the most appropriate design to use given those constraints that I've just described? What are the outcome measures? Liam's just spoken a little bit about that, but how do we assess the effectiveness of the advanced therapies that we've delivered? And then I want to talk a little bit about participant inclusion and some of the limitations surrounding that. So just to talk firstly about trial design. Most people will be familiar with the randomised placebo-controlled double-blind clinical trial. It's the mainstay of pharmacotherapy, even in HD. And it's referred to as the gold standard. But why is that? So the process of randomisation allows us to minimise both selection and allocation bias. The double-blind nature of the trial, where neither the participant or the person measuring those assessments know what group the participant has been allocated to, means that we can reduce both observer and performance bias. And then by adding in a placebo control, we can be confident that any functional benefit we observe in the intervention group over that control group is a result of that intervention itself and not from some other confounding factor. So as I said, the RCT is a great design because it reduces bias in all of these areas. But why might it not be the best design to use for advanced therapies in HD? So the RCT design requires relatively large numbers. I mean, this will differ between trials depending on the primary outcome measure of choice and the variability within that, in that measure. And the introduction of that placebo control, or when we're talking about surgical procedures, a sham surgical procedure, a sham control, is still rather ethically contentious. So a sham procedure is one where all of the aspects of surgery are conducted in the control group right up until the point of intervention delivery. So for the delivery of cell and gene therapies, that means a long, prolonged period of anaesthesia in the participants. And it still remains sort of ethically contentious. Currently, it's mandated by the FDA that we include sham controls in our trials. This is not advocated by the EMA. So even the regulators have a difference of opinion. And interestingly, a recent meta-analysis of cell therapy trials in Parkinson's disease, where they compared sham-controlled trials to open-label trials, found that actually the inclusion of a sham control conferred no methodological benefit. And really, we need to be learning from that data in Parkinson's when we're looking at trial designs in HD. So what are the alternatives to the RCT? One particular alternative, I don't have time to go through them all, is the use of a trial within a cohort. So this is where you have a prospective cohort of participants who have provided consent for longitudinal assessment. Crucially, at the point that they are consented into the cohort, they also provide consent for randomisation to any future treatment without knowing what that treatment might be. So then when you have an intervention that you want to trial, you randomise your eligible participants from that cohort to either receive the intervention or remain in the control arm. The participants who are randomised to the intervention are then informed of their allocation, that they're going to get the treatment, and they're asked to provide consent for that treatment at that point. This is where they can say, no, I don't want to go forward with that. So why is the trial within a cohort potentially a really useful design for advanced therapies in HD? So randomisation of eligible participants from the cohort allows us to have balance and minimise bias between the groups. Because the control participants do not necessarily know that they are actively providing control data for that trial, we can attenuate performance bias, at least in the control group. And through the simple use of devices such as head coverings, so asking participants to wear a hat or a scarf on their heads so that the person doing the assessment can't tell if they've had neurosurgery or not, we're able to maintain a single blind to reduce observer bias. But the trial within a cohort doesn't come without its caveats. You can get increased dropout in the intervention group, which can lead to imbalance between those groups. And it does require an in-depth knowledge of the clinical status of your cohort to determine eligibility. But I don't think that we have that problem in HD because typically our cohorts are very well phenotyped. And actually, as we've gone on and talked about clinical trials over the last couple of days, there's such an appetite for disease-modifying interventions that perhaps we're not going to see the kind of dropout rates when participants are randomised to the intervention that we've seen in other care conditions. So what I'd like to sow the seed of an idea with the audience today is, are there existing platforms, perhaps such as one as Enroll HD, where we could exploit that cohort to function as the cohort for a trial? Now I want to go on to talk a little bit about outcome measures. So Liam's already mentioned sort of the choice of outcome measures for deciding whether or not our ATMP has worked. So the choice of outcome measure will always be determined by the objective of the trial. Early phase trials will typically focus on safety and tolerability, where effectiveness trials will focus on some kind of measure of disease progression and function. Ideally, a good outcome measure is one that is objective, it's easy to assess, and is relatively inexpensive. But what do we include? As we said, we need an imaging measure to make sure that our ATMP is where it should be, and particularly for cells, that they're still alive and that they're integrating and functioning in the brain. We've heard over the last couple of days about the importance of biomarkers functioning as surrogate clinical endpoints. And then we want measures of the disease itself, so across the constructs of motor, cognitive, and behavioural function, and some measures of quality of life. We should also be making sure that we are including outcome measures that are important to participants. What do people with HD want to get from these potential treatments? What kind of improvements do they want to see in their daily life that means something to them, and how do we measure those? But I think the point I want to make here as well is that for all of these constructs, we have a huge number of potential outcome measures, and how do we choose which to use out of those? The other thing that we need to consider is, when do we measure these? At what point after intervention delivery do we want to start measuring cognitive, motor function and behaviour? The point here being that repeated measures are time-consuming, they're expensive, and also, importantly, really burdensome for participants. So we need to make sure that we are employing our outcome measures in a really efficient manner. So this leads me on to mention the COMET initiative. So this is a methodological initiative, and it's a framework for the development of core outcome sets. So a core outcome set is a standardized set of outcomes agreed by consensus with a range of stakeholders that should be reported as a minimum in a specific healthcare area. And what we are advocating for is the generation of a core outcome set for advanced therapies. This is not only important in reducing research waste, so we're not measuring more than we should be during our trials, but by having a core outcome set, it means that we can perform meta-analyses more simply. So our thinking is that with advanced therapies and HD, we're not going to have the typical phase 1, phase 2, phase 3 clinical trial pathway that you'd normally expect from pharmacotherapy, and that actually we're going to have a number of small, iterative, cumulative trials where meta-analyses will be really, really important. And then, if we have this core outcome set, is it possible for us to embed it in a platform such as Enroll HD, so we can get longitudinal readouts of these measures that are important for us to record, so that Enroll HD then functions as a trial-ready cohort from which we can randomise. So I also wanted to talk about how we include people with HD in trials of advanced therapies. As Liam has already mentioned, these trials are going to be run at a number of discrete centres that have access to specialist equipment and expertise, and that typically means that the inclusivity of your participants will be restricted to the region or their co-location with that research site. But really, we need to be broadening our reach to make sure that we provide equity of access to these trials to all people with HD. So we're going to have to go further afield, which leads us on to monitoring. If we are recruiting people who are not local to the delivery centres, we need very specific plans for follow-up and care of these participants. We need to have an open dialogue with their local care teams so that they are informed about the trial, about what's happened and what it means for the participant, so that in the event that anything goes wrong, the participant has a local contact point they can go to reduce their own distress. They don't have to wait for this delivery team in a far-off location to get back to them. And then I wanted to just mention here about communication. So as we heard in the round panel discussion yesterday, managing the hopes and expectations of people with Huntington's when we ask them to join in these trials is incredibly important. And that should be done really comprehensively as part of the informed consent process. But it's not a single point in time. That informed consent process should be dynamic and not static. And it's really important, because of this requirement for long-term follow-up for the safety of the participant, that we maintain engagement and maintain their engagement to adhere to those follow-ups. And maintaining that communication throughout is really important. And lastly, I wanted to talk about the participant voice. It's essential that we include people with HD in all aspects of trial design, outcome measure selection, evaluation of trial processes, and the dissemination of results. And when I say we include people with HD, lone voices are not enough. We need a breadth and depth of opinion to inform the design of our trials. What may be acceptable for one person in one country may not be acceptable for a person in a different region, and we really need to explore that. And I just wanted to end on a personal reflection. I currently co-lead a study with my colleague Emma Lane in Cardiff called the Learn Study, where we talk to people with neurodegenerative disease who've gone through a clinical trial that has involved neurosurgery, with the aim being that by listening to their experiences, we can improve trial designs. And although I have been a proponent of public and patient involvement for the last 10 years and have actively involved the members of the public and participants in designing research, it's still very easy for me, because I'm divorced from the clinical interface, to see participants in a trial as a number on a spreadsheet. But having listened to hours of people telling their own stories, it's really hit home that each one of those numbers is an individual with their own story. They have their own hopes, beliefs, and motivations. They have their families with those same beliefs, hopes, and motivations. And what we need to be doing as researchers is developing patient-centered trial designs to make sure that we are creating trials for people with Huntington's disease, not about them. And that just leaves me to say thank you, and to say that all of the concepts that I've talked about today have been born out of conversations that we've had as part of the Stem Cells for HD and Advanced Therapies working groups. Thank you.